Purkinje cells, or Purkinje neurons, are a class of GABAergic neurons located in the cerebellum. They are named after their discoverer, Czech anatomist Jan Evangelista Purkyně. Anatomy. These cells are some of the largest neurons in the human brain, with an intricately elaborate dendritic arbor, characterized by a large number of dendritic spines. Purkinje cells are found within the Purkinje layer in the cerebellum. Purkinje cells are aligned like dominoes stacked one in front of the other. Their large dendritic arbors form nearly two-dimensional layers through which parallel fibers from the deeper layers pass. These parallel fibers make relatively weaker excitatory synapses to spines in the Purkinje cell dendrite, whereas climbing fibers originating from the inferior olivary nucleus in the medulla provide very powerful excitatory input to the proximal dendrites in cell soma. Parallel fibers pass orthogonally through the Purkinje neuron's dendritic arbor, with up to 200,000 parallel fibers forming a granule cell Purkinje cell synapse with a single Purkinje cell. Each Purkinje cell receives Ca 500 climbing fiber synapses, all originating from a single climbing fiber. Both basket and stellate cells provide inhibitory input to the Purkinje cell, with basket cells synapsing on the Purkinje cell axon initial segment and stellate cells onto the dendrites. Purkinje cells send inhibitory projections to the deep cerebellar nuclei, and constitute the sole output of all motor coordination in the cerebellar cortex. Origins. There is evidence in mice and humans that bone marrow cells either fuse with or generate cerebellar Purkinje cells, and it is possible that bone marrow cells either by direct generation or by cell fusion, could play a role in repair of central nervous system damage. Further evidence points yet towards the possibility of a common stem cell ancestor among Purkinje neurons, B lymphocytes and aldosterone-producing cells of the human adrenal. Electrophysiological activity. Purkinje cells show two distinct forms of electrophysiological activity. Simple spikes occur at rates of 17/150 Hz either spontaneously or when Purkinje cells are activated synaptically by the parallel fibers, the axons of the granule cells. Complex spikes are slow, 1/3 Hz spikes, characterized by an initial prolonged large amplitude spike, followed by a high-frequency burst of smaller amplitude action potentials. They are caused by climbing fiber activation and can involve the generation of calcium-mediated action potentials in the dendrites. Following complex spike activity, simple spikes can be suppressed by the powerful complex spike input. Purkinje cells show spontaneous electrophysiological activity in the form of trains of spikes both sodium-dependent and calcium-dependent. This was initially shown by Rodolfo Linares and Sugimori P-type calcium channels were named after Purkinje cells, where they were initially encountered, which are crucial in cerebellar function. We now know that activation of the Purkinje cell by climbing fibers can shift its activity from a quiet state to a spontaneously active state and vice versa, serving as a kind of toggle switch. These findings have been challenged by a study suggesting that such toggling by climbing fiber inputs occurs predominantly in anesthetized animals and that Purkinje cells in awake-behaving animals, in general, operate almost continuously in the up state. But this latter study has itself been challenged and Purkinje cell toggling has since been observed in awake cats. A computational model of the Purkinje cell has shown intracellular calcium computations to be responsible for toggling. Findings have suggested that Purkinje cell dendrites release endocannabinoids that can transiently downregulate both excitatory and inhibitory synapses. The intrinsic activity mode of Purkinje cells is set and controlled by the sodium-potassium pump. This suggests that the pump might not be simply a homeostatic, housekeeping molecule for ionic gradients. Instead, it could be a computation element in the cerebellum and the brain. Indeed, a mutation in the Na +K+ pump causes rapid-onset dystonia Parkinsonism. Its symptoms indicate that it is a pathology of cerebellar computation. Furthermore, using the poison ouabain to block Na +K+ pumps in the cerebellum of a live mouse induces ataxia and dystonia. Numerical modeling of experimental data suggests that, in vivo, the Na +K+ pump produces long quiescent punctuations to Purkinje neuron firing. These may have a computational role. Alcohol inhibits Na plus K plus pumps in the cerebellum, and this is likely how it corrupts cerebellar computation and body coordination. Molecular profile: the Purkinje layer of the cerebellum, which contains the cell bodies of the Purkinje cells and Bergman glia, express a large number of unique genes. Purkinje-specific gene markers were also proposed by comparing the transcriptome of Purkinje-deficient mice with that of wild-type mice. One illustrative example is the Purkinje cell protein 4 in knockout mice, which exhibit impaired locomotor learning and markedly altered synaptic plasticity in Purkinje neurons. PCP4 accelerates both the association and dissociation of calcium with calmodulin in the cytoplasm of Purkinje cells, and its absence impairs the physiology of these neurons. Medical conditions related to Purkinje cells: in humans, Purkinje cells can be harmed by a variety of causes: toxic exposure, for example to alcohol or lithium. Autoimmune diseases. Genetic mutations causing spinocerebellar ataxias, Unverricht-Lundborg disease, or autism. And neurodegenerative diseases that are not known to have a genetic basis, such as the cerebellar type of multiple system atrophy or sporadic ataxias. Some domestic animals can develop a condition where the Purkinje cells begin to atrophy shortly after birth, called cerebellar abiotrophy. It can lead to symptoms such as ataxia, intention tremors, hyperreactivity, lack of menace reflex, stiff or high-stepping gait, apparent lack of awareness of foot position, and a general inability to determine space and distance. A similar condition known as cerebellar hypoplasia occurs when Purkinje cells fail to develop in utero or die off before birth. The genetic conditions ataxia telangiectasia and Niemann-Pick disease type C, as well as cerebellar essential tremor, involve the progressive loss of Purkinje cells. In Alzheimer's disease, spinal pathology is sometimes seen, as well as loss of dendritic branches of the Purkinje cells. Purkinje cells can also be damaged by the rabies virus as it migrates from the site of infection in the periphery to the central nervous system. References. External links: Cell-Centered Database, Purkinje, Disorders of Cerebellum, ANIF Search, Purkinje cell via the Neuroscience Information Framework. Further reading: Lena S.R., Hess R. Tetrodotoxin-resistant dendritic spikes in avian Purkinje cells. Proc. Natal. Acad. Sci. USA 73, 2520 €3 doi, 10.1073/FNAS 73.7.2520. PMC 430632. PMID 1065905. Lena S.R., Sujimori M. Electrophysiological properties of in vitro Purkinje cell somata in mammalian cerebellus slices. J. Feisel 305, 171 €95 doi, 10.1113/Gisel 1980 SPO 13357. PMC 1282966. PMID 7441552. Lena S.R., Sujimori M., Czech B. Voltage-dependent calcium conductances in mammalian neurons. The P-channel. An. N.Y. Acad. Sci 560, 103 €11 doi, 10.1111 per Joule 1749-6632.1989 TB24084X. PMID 2545128. Forrest, Michael. Biophysics and Computations of the Cerebellar Purkinje Neuron. CreateSpace. ISBN 978-1500245454546.